This is a very important question because we have two different settings in order to design innovative clinical trials in breast cancer. We have a neoadjuvant setting, and then we have a metastatic setting. So in the neoadjuvant setting, we have finally three types of innovative clinical trial design. The first one is the neoadjuvant pure setting, in which you enrich the population according to a specific biomarker. So just to understand better, you can enrich according to HER2 positive, or according to triple negative, or according to ER positive. In the neoadjuvant setting, you have also the residual disease clinical trial design. In this case, you expose patients to the neoadjuvant treatment, and then you enrich the population with residual disease that we know very well that are high risk. There are many trials designed in the post-neoadjuvant setting. Just remember the CREATE-X with capecitabine in the post-neoadjuvant setting for triple negative disease, and the KATHERINE trial that will be presented during ESMO Breast in HER2 positive disease with residual disease following neoadjuvant anti-HER2 therapy plus chemotherapy, and the patients have been randomized to T-DM1 versus trastuzumab. Finally, we have the adaptive trials. In this case, you select a specific biomarker, you explore the activity of a specific agent targeting the biomarker. If you have activity, you expand the trial. If you don't have activity, you stop the trial and you explore another biomarker. Regarding the metastatic setting, of course, it's quite important to remember that the majority of the trial of the future will be designed according to the presence of a specific biomarker. So breast cancer is no more one single disease, but is fragmented according to the presence or not of multiple biomarkers. Now we have many new drugs approved for breast cancer. All these drugs are approved in the context of clinical trials, where you have a very good selection of patients. So patients included in clinical trials are selected according to the presence of a specific target, and also they are selected without comorbidities. So once we move in the real world, we do an extrapolation. It means that patients treated in the real world are not the same of those treated in the clinical trials. So we need to generate in the futures much more data in the real world, because in a clinical trial, you test the efficacy of a drug. In the real world, you will test effectiveness. And so collecting real world data will give us the opportunity to have big data set where you can really confirm the efficacy of a specific drug.